All right, so before we start our seminar, Mario has some announcements to make. So Mario?
Yes, magandang umaga po sa inyong lahat. Magandang gabi din for those who are here in the US. I am Mario Santo Domingo, the president of the Philippine American Academy of Science and Engineering, and I would like to welcome everyone to our webinar featuring Professor Eric Bejarano. He'll be introduced later by Dr. March Peña, our moderator. At this point, I'd like to also recognize the presence of our PAASE members, of course, Vice President Gladys Completo, Dr. Elmer Mojica, and yeah, of course, everyone who is currently here online with us. So I just would like also to invite everyone to our future webinar. This is next week, or fireside chat, or rather, on the UP Resilience Institute's efforts towards resilience building, featuring Dr. Alfredo Mahar, Francisco A. Lagmay from the National Institute of Geological Sciences, and of course, the University of the Philippines Resilience Institute. It will be moderated by Dr. Cleto Ting Nanyola. I think he's here also with us, right? And the reactor will be Dean Felino Lansigan. I also would like to invite everyone to the 42nd annual PAASE meeting and symposium to be held on October 11 to 14th, 2022. This is spearheaded by our colleagues from the University of Santo Tomas. If you want to know about the details of the APAMS, you can visit apams2022.paase.org, and you can also see there the links to abstract submission and to the registration. Before we begin, I also would like to show you two short clips, after which Dr. March Peña will start the session.
There's no sound, Mario.
March didn't want to play, so we can we can continue.
Okay, it's what did you want to do the
Sayang J. We can play it later because it's
okay, we'll do it later. Okay. All right. Okay. Well, good evening, everyone in the US. Good morning, everyone in the Philippines. Welcome to tonight's webinar. It's my great pleasure to present our speaker today, or to introduce our speaker today, Dr. Eric Bejarano. He is a newly tenured associate professor here at the University of South Carolina at the Center for Environmental Nanoscience and Risk at the Department of Environmental Health Sciences, University of South Carolina. Eric actually received his BS in agricultural chemistry from the University of Philippines in Los Baños. And of course, many of you here are from Los Baños. He got his PhD in chemistry from Louisiana State University. Along with his PhD mentor, he contributed to pioneering work on environmentally persistent free radicals, also known as EPFRs. After his graduate studies, Eric completed his postdoctoral training at Department of Civil and Environmental Engineering at Virginia Tech, where he worked at one of the leading air quality labs in the United States. In 2017, Eric was among the 30 faculty fellows of what was then the newly launched National Science Foundation EPSCoR2 R2 Track 4 initiative, the goal of which was to increase the number of researchers, the next generation of researchers in the United States. In 2022, he received the prestigious National Science Foundation Career Award, aptly cementing his recognition as one of the outstanding young investigators in the United States. He has collaborated with scientists at the National High Magnetic Field Laboratory. And among his contributions is the understanding of the behavior of bioaerosols, which informed the contents of the CDC prevention's website on COVID-19. So a lot of his work was highlighted by the publication by the Directorate on General Environment by the European Commission. And basically it is designed to inform policymakers about the latest environmental research findings that are designed to design, implement, and regulate effective policies across the European Union. So without further ado, I would like to introduce Dr. Eric Bejarano, who will be presenting his talk on particle chemical interactions and their implications. Eric?
Okay. Thank you, Ate Marge. Good morning, everyone in the Philippines, Dito America. First of all, this presentation will basically be just a smorgasbord of research that I have done since my graduate studies and in my postdoc and currently now at USC and my future research direction. So first of all, I'd like to thank the funding agencies, especially the National Science Foundation, the National Institutes of Health through the NIEHS, and the United States Army, who funded me for the last 5 years or 6 years. And to Professor Barry Dellinger, he was my PhD advisor, and Professor Linsumar, my postdoctoral advisor at Virginia Tech, and especially to these two students, Dr. Rao and Dr. An, for helping me a lot with this tenure process. So to start with, my talk would be about particle chemical interactions and their implication. And particles are very important components of the atmosphere. When you look at the atmosphere, 80% of the surface in the atmosphere is actually due to particles. So some people call it aerosol. We're going to use it interchangeably in this talk. So air pollution is a leading death, global cause of death, at least. That's according to the latest finding, and it has been implicated with some of morbidity factors, cardiovascular diseases, and part of it because particles, depending on its size, are very hazardous, aside from its chemical composition. So growing up in the Philippines, I think until second year high school, I studied under a lamplight. So this picture connotes so much emotions when it comes to studying air pollutants, and especially from combustion sources. So my talk will be about combustion sources. So And I'm going to focus here on nanoparticle first. And when we talk about nanoparticles, our ability to actually produce nanoparticles or our exposure to them is not new. It is as old as humans, our human story. And when our early ancestors discovered how to create fire, they had been inadvertently creating nanoparticles, and they have used it as part of this a wonderful mural in the Chauvet Cave in France. But what is unprecedented today is that we have the ability to control the functions and modify this nanoparticle depending on our applications. But coupled with that, our exposure to nanoparticles and hazardous particles in general has been dramatically increased over the last centuries due to anthropogenic human activities. So for those who are not familiar with nanoscience and nanotechnology, a nanoparticle is defined as a particle with a size of 100 nanometer, at least in one dimension. So you can have a 1 meter long strand of particle, but if the dimension at least on the length side is 100 nanometer, then it's considered a nanoparticle. It's very difficult though to produce such types of nanoparticle. Now, to put this into perspective, if you have a nanoparticle, let's say you have that blue sphere as a nanoparticle, if you put this into a soccer ball and you try to find out where is that blue dot on that soccer ball, that is analogous to finding that soccer ball on the surface of the earth. So that's how small nanoparticles are. So due to this size difference, it possesses unique size-dependent properties. So if you have a dog and you have somehow magic ability to make this smaller into the nanoscale regime, it will become a cat tweeting like a bird. So this is just the usual description I give to my students about nanoparticles. So you're familiar with gold. Gold is on this side, it's yellow, but gold in the nanoparticle regime would actually exhibit different color depending on its size. So for this discussion, we will discuss the contribution of particles on the formation of persistent free radicals. So these are incidental nanoparticles. These are inadvertently produced as a result of combustion byproducts or thermal processes. And the second one is how engineered nanomaterials are affecting pollutant emission. And the third part of this talk will be about the impact of relative humidity to natural nanoparticles. And I consider viruses as a natural, almost semi-intelligent nanoparticles. And the last one would be my research direction, in the hope that this presentation would result in meaningful collaborations to those who are have overlap with my research in the Philippines and those in the United States. So my first talk is about environmentally persistent free radicals. It's a bit a mouthful, so we just use the word EFRs for short. So everyone knows that a free radical is a molecule or element containing at least one unpaired electron. And because of this unpaired electron, radicals are very reactive. Now, most atmospheric free radicals, like this small hydroxyl radical or the hydrogen atom, and the superoxide anions, they decay really quickly. So they have a life of nanoseconds. And for bigger radicals like cyclopentadienyl, it has a longer lifetime. So as you can see that as you increase the size of the radical, it becomes more stable. Now, what is unique is that we have discovered that environmentally persistent free radicals could last for like hours, which is unheard of for free radicals. Now, this discovery is not new. It was reported back in Nature in 1952. They found that persistent paramagnetic signals come out from combustion of coal, but with that study, it ended there. So it wasn't brought to the interface of environmental aspects, so people did not put a lot of attention into what these radicals are. So back in 2005, my advisor, Professor Dellinger, studied research and extended this to environmental domains. And these are the electron paramagnetic signal when you detect a free radical. So when you sample, let's say, cigarette smoke, or if you burn candles, if you collect the particles there, most likely it would have this signal that is indicative that you have free radicals. Now, these are different cities in the United States. So Baton Rouge, that's where LSU is. It's probably among the dirtiest cities I've been to in the United States, and contains a lot of free radicals. Now, the radical here on the y-axis, that's the concentration in spins per gram. And then here on the x-axis, that's the day of storage. You can see that despite this almost exponential decay, it's actually not really exponential, because when you look at the time scale, it's still 10 to the 17. So this difference is almost negligible. So this tells you that these free radicals are indeed very persistent. Now, the free radicals, EPFRs, are most likely from combustion origins, but when you burn anything, if you even have a candle, it will produce like hundreds, if not thousands of compounds as part of the emission byproduct. So it's difficult to determine what are the components of a free radical. So we go to the lab and try to do a chemical model, a very simplistic model of a metal oxide that might produce, that will produce a nanoparticle, and put it on a big support, and then use an organic molecule to generate the free radical. So these organic compounds, they're mainly chlorinated aromatic hydrocarbons like benzene or phenol. Then they are converted to free radicals. Now, for a free radicals to be called an ethers, at least by the definition that was given by Berry, is that it has to have two characters. So it's a bit cut off here. So the first one is that the free radical should be stable, meaning that once it formed in the combustion, it has to stay the same. It should not degrade. And the second one is that it should be persistent, meaning that it should stay for at least more than the lifetime of those in the atmosphere, like about a second. So if you think of EPFRs, it would have a lifetime that of a tortoise, and those that are like hydroxyl radical, which is the primary oxidant in the atmosphere, which are very important in the chemistry of the atmosphere, would have with an equivalent life of that of a fly. So because of this, EPFRs are hazardous because their persistence enables them to be transported over long distances, and that they can exert their hazardous effects on humans. So from which component of in the combustion or compounds are they formed? So in combustion, there's usually three classes of chemicals. We have organic species, so these are carbon compounds. We have inorganic species, ammonium, sulfate, what else? Carbonates and sulfates, and then we have transition metals. Now the detection of the free radical, they are being suggested it's organic. So we thought that this might be that if you synthesize organic species on a metal, then they form free radical, and indeed they do. So just to give you a simplified version of what's happening in the combustion is that the combustion system is actually defined into zone. It means that when you burn a candle or you turn on your oven, the first step that's happening when you're burning the gas, it's vaporizing, and then there are molecular reactions happening there, producing, resulting to high temperature dissociation. So this will occur at greater than 1200 degrees Celsius, in which many incinerators, at least in the United States and Europe, the temperatures are set on. So the third one is that as the combustion progresses, it cools down. Now, free radicals are formed in the cool zone of combustion below 600 degrees Celsius because they rearrange, the small molecules in zone 2 would rearrange to form slightly larger species to form these free radicals. Now, this is the conceptual mechanism of free radical formation. It's only conceptual because nobody has figured out what is really the mechanism of free radical formation. If you do, you might get a publication in Science. That's what one of the professors there said. So the first step is that an aromatic molecule with a substituent like chlorine or hydroxyl functional group can react on a surface. In this case, we have iron oxide. You can see here that there's OH there because most surfaces would contain water. And that water are not easily removed from the surface of nanoparticles, even at 500 degrees Celsius. They are really stubbornly held on surfaces. So after that, the free radical forms a chemisorb species in which you have chemically bonded aromatic ring by the condensation of the elimination of hydrogen chloride and or water. So depending on your compound. And then succeeding electron transfer would form the free radical and reducing your metal. So that's how you form EPFRs. That's what at least we thought, and from some of the lines of evidence from other studies like IR or XPS. Now, in terms of the persistence of the free radical, so this is the relative radical concentration, and we just track it through time. So this is a pseudo first order rate decay, rate for the free radical for different, this is for zinc oxide with different aromatic precursors. So these are like different compounds that you supposedly burn. So what happens that if you use, it depends, the lifetime of the radical depends on the metal, okay? So here we have relaxation time. So that's a measure of how fast the radical is decaying. So the relaxation time is simply the fold time, which means that what is the time needed for approximately 37% of the free radical to decay from its original concentration. So for copper oxide, we have about 74 minutes. So that's over an hour. So that's really persistent. And if you change your metal to an iron oxide nanoparticle or nickel oxide, it would survive there and remain there 30% of the original concentration for 5 days. And if you produce zinc oxide, use zinc oxide, it would last for 73 days, so more than 2 months. And this lifetime translates to over a year for free radicals and zinc oxide. So the EPFRs is a big thing at LSU. They had been funded by NSF through the Superfund program. They're primarily focused on health effects. And not so much, in my case, I'm more on the chemistry and the physics of the formation of the free radical and not so much of the health effects, which LSU does take care of it. So the reason for free radicals to decay fast is that some of the radical would actually decompose to cyclopentadienyl radical, but then this radical is still persistent though. So depending on the free radical that you form, that would tell you how persistent it would be. So if you have a 7-quinone radical, they're generally more persistent than a corresponding phenoxyl radical. Now the G value here is just to identify the free radical. So that's basically the quantity. It's that, you know, electrons are affected by their chemical environment. And the G factor is the coupling between the spin orbit and the orbital momentum of that electron. So it depends on the chemical environment. So therefore, you can tell at least, can classify them to big classes of free radical. Now, the G value is very limited. So it's like you're in a room, you're trying to find a person, and all you have is a description of like, she's a girl. And that's all you can tell, and you cannot find. There's like 10 girls in that room, so it's impossible for you to tell. We tried to use the fancy state-of-the-art high magnetic EPRs at the magnetic lab, at the mag lab, but we're not successful in determining the structure of free radical. So this assignment is thus more arbitrary. Okay. So in terms of health implication, if they've quantified the free radicals in particulate matter in some cities, and they did a study in which they converted it to cigarette smoke equivalent. And based on the National Ambient Air Quality Standards in the United States, which is about 15 micrograms per cubic meter, in Metro Manila, the amount of particles in the air would exceed that. And in China, that could go to about 20 times. So for free radicals being inhaled, so that's equivalent to smoking 10 cigarettes per day, compared to only one cigarette for 2 days in the United States. So that makes it a very good, what do you call this, studies to determine what are the health impacts of free radical. So we know that free radicals are causing at least some dysfunction, at least in murine and in murine models, mice and rats, at least in the lab. So as a chemist, we look for what is the mechanism by which EPFRs are producing, are causing toxic effect. So the first one is that EPFRs would ultimately decay. They're very stable, but they are not really that stable. Ultimately, they will decay to its molecular form. And the main sink for EPFRs in the atmosphere, it's its reaction with molecular oxygen. So the oxygen then becomes a superoxide anion radical. And in the human body or in biological system, this superoxide will dismutate in the presence of water, producing hydrogen peroxide. When you have hydrogen peroxide, it will react or participate in Fenton reaction or Fenton-like reaction and produce hydroxyl radical, which is the most oxidizing of all these free radicals. Now, in the human body, there are some iron sulfur cluster protein, and these can participate in this Fenton reaction. So we verify this in the lab, if that's producing OH radical. And here's the y-axis, that's the concentration of OH generated. And the x-axis, that's the exposure time. And we'll see here that when you have EPFRs, you have higher, significantly higher EPFR concentration than that of a control or from a sample that do not contain EPFRs. Okay. So this tells us that in terms of the damage, if you have 1 mole of EPFRs, it will produce 10 times hydroxyl radical. So this might explain the observed effect in toxicity studies from the LSU Health Sciences Center. So to summarize this, that the 3 glaring summary for this one is that radicals form on transition metal oxide surfaces and that they're very persistent. And for the human standpoint and NIH implications that they catalytically generate ROS, hence those observed toxicity effects. EPFRs have been implicated with cardiovascular diseases, arrhythmia. And also, if you are exposed to high quantities of EPFRs, you are more likely to be susceptible to being infected with influenza. So the second part is that the work on engineered nanomaterials. When President Bill Clinton signed in 1999, around that time, the National Nanotechnology Initiative, everyone's just hyped with nano particles and putting them on consumer products, making promises that if you drink, put this on your sock, it won't be sweaty, or this magical nanoparticle you put on your beverage would make you, will be healthy. So this is a inventory that we did together with our collaborator at the Woodrow Wilson Center in Washington, D.C. And it shows that since 2005, there's a steady increase in consumer products that incorporates nanomaterials. But after studies that shows that some of the nanomaterials would actually have toxic effect, then I think if you follow this line, this would go down. They did not advertise that their product would contain nanomaterials. Now, these are the most common nanomaterials that are produced and incorporated in many products on different industries, automotive, energy and environment, coating and paints, personal care products, and titanium dioxide and zinc oxide and silica are among the highest. So this one basically tracks how the nanomaterials would flow into the environment. And to this discussion, what is important for this is that the amount in the solid waste incineration plant. So that's the wastewater plant, and only a small fraction of the nanomaterial will eventually end up being incinerated. And when you incinerate, only a fraction of them would actually be emitted into the air. Now, this study, I think, overestimated that, 8,100 out of 86,000 that would go to the incinerator would be emitted in the air. That is not quite what we found in this study. So the point of this is that if you have nanomaterials in consumer products or in solid waste, what happens? Does that affect pollutant emission? And do the nanomaterials affect the emission of combustion byproducts? And if so, does it also change the aerosol properties of the emitted particle coming out from the exhaust? And if the nanomaterials themselves changes its behavior and properties as a result of being incinerated, and that's important because changes in nanomaterials property might exacerbate its toxicity effect. Now, the thing about nanomaterial, unlike chemical compounds that are toxic, is that if you have, let's say, benzene, which is carcinogenic, if you put benzene in soil, benzene in air, benzene in water, it would still be the same, have the same effect. But nanomaterials changes a lot because the surface property, the size, and the surface charge, and the coating changes the aspect of its toxicity. Now, so we went to the lab, just add nanomaterials and unleash your inner pyromania. So I basically just keep on burning things in the lab and then measure changes on them. So the first one is on pollutant emission. So we track polycyclic aromatic hydrocarbons. But these are, if you like barbecue, I'm sure there's a lot of PAHs there. If you burn wood, there's a lot. Now, PAHs especially the 16 species of PAHs are among those that are tracked in many pollution emission studies. Now, with PAHs, so this is naphthalene, which is in mothballs. And what happens that when you emit them from combustion, the bigger PAHs, which are usually the carcinogenic ones, tend to absorb on particles. So they piggyback on particles, and those in the gas phase are mainly the smaller particle. Okay. So now, so this is the result of the emission studies. I know this is a very busy graph, but we'll just focus on a couple of things here. So the y-axis here, that's the emission factor. Now, the emission factor here is simply defined as the amount of pollutant being emitted relative to the mass that you're burning. And these are the 16 PAHs. And the different colors here are just for the different nanomaterials that were added into those solid waste like paper, plastic, and PVC. And you can see here that there are three main PAHs that actually stood out of this graph. And also there's this, what do you call this? This clustering here for chrysin, which is a bit larger polycyclic aromatic hydrocarbon. And then you have benzobifluorantene when you add ceria. Now, back in the days, I'm not sure if they still add ceria now in automobiles. Ceria is added on the engine to reduce particle pollution. But from the result of these studies, that when you add ceria, you actually enhance the formation of this 5 to 6 ring PAHs, which are, some of them are carcinogenic. Okay. So the other one, so we compare now the three PAHs here are emitted, and the reason for them being the most dominant in combustion is that naphthalene is formed from 2-cyclopentadienyl radicals. So you only have this basic structure to actually recombine, and then you would get these three structures that we actually found there. Now, we want to know how does the emission factor or emission of the nanomaterial differ from that of the bulk. So if you add bulk gold, let's say you have lots of money to burn and you can put some chunks of gold there that are really big compared to nano gold, which is still expensive, but not very expensive. So what would be the difference? So there's two things that we notice is that first, the emission factor are a lot different. For those with bulk nanomaterial in which you add big silver chunks or ceria or titania, the emission is actually lower compared when you put nanomaterial on the sample. And the second one is that the most abundant PAHs for the nanomaterials, when you add nanomaterial, is naphthalene and fluorine. and the other one is phenanthrene. But overall, they only emit mostly small PAHs, which is good because these are only suspected carcinogens, at least some of them. Now, we also track dioxin emissions because they're among those that are tracked. And to our surprise, we did not detect any dioxin. Now, one reason is that dioxin is normally formed through the elyridal mechanism in which you have to have a radical on the surface. So that's on a nanoparticle. And then you had to have a compound in the gas phase, like here phenol. Now, the phenol cannot approach because this is, of course, an oversimplified picture of what's happening there. But the surface of the silver, for instance, of the silver nanoparticle is really crowded, so that steric effect could inhibit this OH from interacting with the free radical that is on the surface. But furans actually were emitted at very high concentration. So we have 2.5 times 10 to the 4 picogram per gram. So this was done with a high resolution GCMS. And because free radicals are formed, furans are formed via the Langmuir-Hinshelwood mechanism, in which you need two more radical on the surface to form. Now, the kinetics might be slower because they're absorbed on the surface, but at least if you wait long enough, it's thermodynamically favorable, it will form furans. Now, when you put this nanomaterial, it changes the number of the properties of the particle or the aerosol being emitted from the combustion system. And most people, it's a misconception that very small particles are actually hazardous. Now, when you hear about that, you have to qualify what is meant by small. Now, if you have, let's say, 5 nanometer particle, the perspective for human inhalation, that is not very dangerous. And the reason being is that the small particles are actually deposited here on the upper respiratory tract, because the small particles like 1 nanometer, 5 nanometer, tend to have high diffusivity, meaning that Brownian motion, air molecules colliding on the nanoparticle pushes it away so that your nose hair is enough to actually filter those particles. Now, the more dangerous particles are a bit larger, 30 nanometers, 100 nanometer. They would actually the ones that would go into the deeper part of your lungs. Now, in terms of aerosol size, so the y-axis here is the particle median diameter. So that is just the size of the particle. And when we add a different nanoparticle and track how big is the particle coming out of the incinerator. So this one here is for the control, and the red one here is, oops, not moving. So the red one here is for the 10 weight %. So this is the loading of the nanomaterials that we put. Okay, so we change the amount of nanomaterials. We put about 1x, and then we multiplied it 100 times and see if there are any changes. Now, sure enough, when you put a small amount of nanomaterials, there's almost no change relative to a control in which we did not add any nanomaterials. But when you add 100 times, the nanoparticle decreases to 300 nanometer. Now, that is a very interesting result because the 300 nanometer particle is actually the gold standard for measuring particle filtration efficiency, because the 300 nanometer particles are very difficult to capture because they're not big enough to sediment by gravity, and they're not small enough to be bombarded to exhibit Brownian motion effect, but thus they can actually pass through filters. So those HEPA filters in your office, those were mainly measured at 300 nanometer particle. Okay, so there's a big change there, but in terms of the amount of particle number, so this one is about 10 to the 13, there's really no impact on nanoparticle number concentration. So even though that concentration is still very high, so this is a very high amount of particle number coming out from the incinerator. Now, we did some tests to check if the nanomaterial is changing. Now, one of the ways to do it is that this nanomaterial most of them are crystalline. So when you do electron diffraction studies on them, you would actually measure the interplanar distance spaces, and these spaces are characteristic of a certain crystalline phase, and therefore you can able to it's like a fingerprint basically. Now for crystalline, you can see these really intense dots. And for amorphous compound, you only have this hazy ring similar to that in Syria. So we perform these studies, and with this one, they're essentially what this images tells us, that there's actually no changes that we observe on those nanoparticles. So which is a good thing, because then the toxicity, their toxicities are not modified, but except for silver and quantum cadmium selenide quantum dots. So we started with this nanoparticle about 5 nanometers, but after combustion, they actually melted and then they fused, then they form highly spherical particle, which is one of the ways you can tell if a nanoparticle is actually from a combustion origin or if it's actually inside the body. So there was a study back then published in PNAS in which they know that there are some nanoparticles in the human brain from brain studies from Mexico, and found that there are iron oxide nanoparticles that are highly spherical, and therefore they are coming outside and from combustion sources. But anyway, to go back to what I'm saying is that this nanoparticle became larger, so which is good, because then the toxicity decreases with increase in size. So only cadmium selenide, quantum dot, and silver nanoparticle here were the ones that exhibited changes in melted during incineration. So by the way, I burned this material at 800 degrees Celsius. And for nanomaterials, since there's actually a size effect with the melting point, if you decrease the size, then the melting point becomes even lower. So that's the reason why the very small quantum dot actually became smaller compared to the larger nanoparticle I used. Now, in terms of the flow of nanomaterials, just for its environmental implication, if you remember what I said previously in previous slides, is that Keller and Lazariva overestimated what's coming out of the incinerator. It's almost like about 90% of the nanoparticles will be emitted into the atmosphere. That is not the case of what we found. It's actually the opposite. More than 99.5% will stay in the incinerator as a bottom ash, and only less than 0.50% will go into particulate matter. And of course, a caveat for this one is that these were done in a lab scale studies. A typical incinerator would have turbulence factor, and that changes a lot. But I would not see it going into that 85 to 90% being emitted. It would be less than that. And for most incinerators in Europe and the United States, you have at least air pollution control devices, then the emission is a lot lower into the air. A lot of them, though, will ultimately end up in landfills. So that's where the bulk of the nanomaterial will go, and not into the air. Now, in the Philippines, where in some places open burning is still happening, so that might be a concern. Now, in terms of the toxicity studies, we use the nanoparticles and what is coming out after burning the material with the nanoparticle. And we use toxicity assay, so mainly a cellular assay, to assess its oxidative potential, because that's the first step in establishing the toxicity. And if the body could not manage that, then it could undergo inflammation and to higher tiers of toxicity effect. So we use cellular assay to test for cell viability using the WST1. And we also tested the impact of if there's really high amounts of oxidation using genotoxicity studies using histone immunofluorescence assay. So what this assay tells us is just if OH is causing the DNA to break, the histone will react on that one and become fluorescent and form foci. And the number of foci that you detect on the cell will actually increases with its toxicity or that DNA damage. So here is more suffered a lot more breakage than, for instance, this small dot here or this isolated dot here. So here, oxidative potential. So I'm only showing glutathione. Now, glutathione in the body, so we have that in the body. About 95% of the glutathione in the body exists as reduced glutathione. Once you have high amounts of stress, oxidative stress, then it gets oxidized to its disulfide form. And therefore, as a measure, if you look here on the x-axis, what we're tracking is that a lower value of the GSH to GSG ratio would actually mean that you have higher oxidative potential. So as a control, we have here hydrogen peroxide, which is highly oxidizing, and therefore there's zero. So almost all of the GSH were converted to its oxidized form. Now, the take-home message for this one is that for some nanomaterials, there's some toxicity effect, but overall they were no different from that of a few of the control. Okay. So here's the control. Here's the control, and some of the nanomaterials were still on that range. Now, the second one is for cytotoxicity. So again, I'll just shortcut with this one. And the take-home message for this one is that in terms of the PM compared to the control, there's not so much difference. When you look at this error graph, there's so much overlap, so these are not significantly different. Okay. Now, for the genotoxicity studies, what we're looking for is that if there's a massive genotoxic effect, then we should see a lot more foci. So these are the dots. And we should look here at the larger number. So if you have 21 to 30 foci on a cell, then that would indicate massive genotoxicity effect. But when you look here, there's not really so much genotoxicity effect. So overall, the conclusion for this study on toxicity is that the nanoparticle doesn't actually modify so much the toxicity of the nanomaterials. What it does modify is the emission of some pollutants, and particularly the aerosol size being dragged to a very dangerous size of particulate size. Okay. Now, going back to, so I work on this research, this next one I'm presenting here at USC. Now, this study came actually from ideas that were generated from the studies I described previously. Now, if you remember when I mentioned about furans being formed with two radicals on the surface, that study, that was more like a hypothetical idea. So it's not that we're measuring EPFRs, because at Virginia Tech, we don't care about EPFRs there. It's more about aerosol. So I brought that there and wondering if they are forming huge amount of furans, there must be a lot of EPFRs that are being formed. So now the difference with this study is that at LSU, they actually use big particles, and on those big particles, you have islands of nanoparticles. And on these islands, this is where you're producing those EPFRs. Now, what would happen if you actually remove the support and you basically only have pristine nanomaterials, nanoparticles? Would they still form free radicals? Now, that's a big question, because it's easier to form here because there is a Schottky contact. So now what it means that there's two materials of different composition, so they have different dielectric constant, and electrons are able to shuttle between those due to those gradients. But if you remove those gradients, would you still form those free radicals? So what we did here is to actually just try to knock down some of the ideas about how EPFRs are formed. Do you really need a support to form EPFRs? And then now for the career award, we are investigating that, do you really need the metal oxide actually to form EPFRs? And that if you have particles that are compounds in the air that are transformed by its atmospheric processes and oxidation, you probably don't need metal oxide. They can form persistent free radicals without this. Okay, so this is the one I mentioned a while ago, and also this study is actually due to to the concept of zinc 2 plus producing free radicals. When I started it at LSU, I was thinking that zinc 2 plus, according to this mechanism, should not form a free radical, because this free radical formation posits on the formation on electron transfer. So you have electrons transferring from oxygen to the copper and reducing it. But when you do it for zinc 2 plus, there's actually no zinc plus oxidation state. When you reduce reduce zinc 2 plus, it will actually become metallic zinc. So, but to our surprise, you form high amounts of free radicals and they're very persistent. So what is causing it to produce that free radicals? And also, would it produce without any support? So with this one, we studied proposed a different models on band bending. So the band bending is essentially just when you have a particle, and particles like a semiconductor would have valence bond and conduction bond, and that if you have a large particle, you need a higher potential to actually cross it, so to cross the barrier, compared to if you have a small particle and you only need a very small energy to surmount the barrier. Now, this is so this is analogous to that. If you fell in a hole and that the hole is very deep, then you cannot get out. If the hole is actually shallower, then you can get out. So the important thing here is this two quantities from this equation. It's very simple, though. It only relates the depletion layer, which is related to the size of the nanoparticle here, and to the relative permittivity constant, which is a property of the material. So what happens here is that we hypothesize that nanomaterials will form free radicals and that small nanoparticles will induce a small band bending or may even be negligible. So these are the claims I made back in 2018 on ESNT paper. And so the second one is that zinc oxide will have higher band bending, so therefore it will not form free radicals compared to titanium dioxide. So with this, we went to the lab and see if these claims are actually true, and indeed, so these predictions are true. We formed on titanium dioxide, if you have 13 nanometer particle and you have 13 nanometer zinc oxide, you do not form free radical on zinc oxide on the basis that zinc oxide has a lower ER, and therefore the band bending is actually a lot higher, and you cannot, and electrons like trying to move around between the surface and the organic compounds could not do it. So here's the EPR spectra for the free radical. You see this very intense signal compared to 1 to dCb, but overall they were forming really intense signal, and that it's not so much about electron reduction, it's preliminary evidence though, because there were no changes on the, at least on the fringe distances for this nanoparticle. Regardless of the condition, whether you use the pristine nanomaterials or you just heat it up or you form EPFRs on the surface, the materials stay the same. It's characterized with this fringe pattern. So now for zinc oxide, so here's the graph. We did not form any EPFRs there, but if you dose the EPR with ethanol, if you doped it, and then it forms this signal. Now, with my collaborators at the Beijing Institute of Technology, they did studies, not really directly related to this, but there's some direct link, some indirect link, is that if you have some compounds like acidic or basic pollutants, there's interaction between the nanoparticle and the organic compounds on the surface, and that this interaction removes the barrier for the free radicals to form. So essentially it removes the hump, and the free radical is it's easier for them to form. So this is the one for another one is that you can actually make the size smaller. So then it confirms our hypothesis that indeed that you can make the particles smaller and it will form free radicals. And even though that zinc oxide is notoriously difficult to form free radical. So it took a while for the grad student to form these free radicals. Now, this is important because titanium dioxide and zinc oxide are among the most put compounds in nanoparticles in consumer products. And with these findings it will inform, whether nanomaterials in terms of their disposal strategy. Can you just burn nanomaterials or you don't have to? And also might be used as a substitute to find other nanomaterials that may be more less hazardous. And zinc oxide is mainly put on a lot of them as, what do you call this one? As sunscreen. Now, the last one here in terms of the nanoparticles, the fate of bioaerosol in indoor air. So this studies was also done at Virginia Tech and is related to the COVID pandemic. But this one was actually done to understand the impact of relative humidity on the influenza virus, especially its seasonality in the United States. So in the US, influenza season is in the start of the fall and until spring. In tropical countries, it's during the wet season, and relative humidity plays a big role in that one. So if you expel respiratory droplets into the air, the concentration of that infectious virus is actually removed by 3 mechanisms. First, big droplet settles by gravity, and the other one, in case of most indoor environments in the US and in Europe and the Philippines, you have ventilation system to take it out, and the viruses itself is actually inactivated a response to the changes in humidity. Okay. Now, the humidity is important because it controls the final size of the particle. When you expel, let's say when you sneeze, before they come out from your body, they're actually at 100% relative humidity. It's really saturated there, but once they exit and into the environment, in the winter, it can go to 20% relative humidity, and therefore the water evaporates and form the droplet. Now, this droplet is dictated by the balance between the Kelvin effect, in which you have a particle and the particle becomes smaller, the vapor pressure of water becomes high, so then it has tendency to evaporate. But then that one is counteracted by Rayleigh law effect, in which if you have non-volatile solute like salt, then the salt will decrease the vapor pressure and make the droplet reach its final equilibrium size. So most of the studies on respiratory droplet has focused on the physics. Now, if you have 10 micrometer, which has been the cutoff for some of this droplet aerosol debate, especially during the start of the pandemic, and Lindsay was among the first one who pushed for the ideas of redefining on showing that COVID is transmitted by the air during the, and they're the ones who were able to establish that it is in the air. Now, my focus on this one is to understand the chemistry, which is less understood. So what's happening as the droplet evaporates and the water gets out of the droplet, it changes the pH, the concentration becomes higher. And what do those events affect the virus? So we studied, water and salt and mucin, which has been usually used for many simulated saliva studies, and we also use added a surfactant, and we also tested human saliva. So this graph, the key conclusion for this one is that for many studies, especially during the start of the pandemic, in which people were zealous to contribute to studies, but some of them are not well designed, and they ended up using just water, sodium chloride, and confusing the literature. But that one would not give you a good picture or a simulation of a respiratory of a saliva. So you have to add surfactant, which has a big impact in terms of mimicking the behavior of human saliva. And from this one, you can see that the level here is about 7 × 10⁻⁴, and this one is also the same, and they almost have similar evaporation rate. So that means that you have to use DPPC in order to mimic saliva. If you only put mucin, which is a protein, by the way, as a big major component of saliva, then it's not going to be representative or mimic the behavior of your droplet. So we did confocal fluorescence studies and on some the pH measurement. So this is a droplet that is at 95% relative humidity. So when you are at 95% relative humidity, the droplet here is opaque. Now, if you decrease the relative humidity to 80%, it becomes transparent. Now, what's happening here is that the mucin, which is the protein that we put, is actually undergoing gelation. So it's getting a bit ordered. So, and also the contribution, it's known that gelation for mucin can occur under high salt concentration, in which here the salt is starting to increase in concentration as the water evaporates. And also there's a change in pH. So this paper here by with other studies we did on the pH of the droplet is that there's actually a gradient in the pH between the surface and those in the bulk. So that translates to about 3 to 4 orders of pH magnitude. So that's about 10,000 times change in the concentration between on the surface in that inside the droplet. Now we track, so this is the studies in which we're trying to see what would happen to those components. So this is important because as the components in the saliva separates or whatever's happening with them, it will dictate the location of the virus. Okay, so the red rings here are actually the mucin protein. So we tag it with a fluorescent dye. And you see that initially they actually concentrate on the surface. So I think because they have, compared to the others, they have low surface tension and they tend to stay on the surface due to Marangoni effect. But as you decrease the relative humidity to by 5% every minute, and you finally settle to 30%, you actually see localization of mucin here. So you have this highly intense red dots here, and here. So that means that the proteins are aggregating on one side and the salt, this is a salt crystal here, is separating from the rest of the droplet. Now the other one is we did the same studies except we added surfactant. Now you don't see the ring effect on the surfactant when you add a surfactant. So we add DPPC, which is the largest surfactant component in the lung. And when you add that one, you don't see that. And also the DPPC is for some strange behavior, actually partitions into the salt rather than spreading outside of the ring. So I would expect that the DPPC would be outside because it has low surface tension and it should be actually be the one on the outside of the ring. So I'll summarize some of this one because I think due to time limitation. This other research is done with the US Army. We try to measure VOC partitioning on particles. Now the Army is interested with this one because in the event of chemical spill or some, what do you call this one? Advertent chemical exposure to contaminants, we wanted to know how the pollutants would move between the soil into the air and those in the air going into particle. So particularly we are interested in this one. This one is rather easy to measure, but the challenge is for this to measure. So we took a lot of time trying to figure out how do we measure partitioning coefficient, which is actually just defined as if you have a pollutant, some of them will go to the particle, some of them will stay in the gas phase. So what's the ratio of those going to the gas phase and staying in the gas phase and those staying in the particle phase? Okay, so that's essentially what it is. And the main point for these studies is that we wanted to know this very basic constant, which is very simple. It's basically your extraction coefficient if you're going liquid extraction in chemistry lab. But the difficulty with this one is that there's tons of there's huge concentration of the VOC of this volatile organic compound in the air compared to the very small amount in the atmosphere. So how do you measure something that is present in small amount and it's clouded by this massive amount? So the the main goal of this study was actually for assessing exposure. So there's not a lot known with VOCs, and VOCs are among the highest, the majority of the pollutants now in the atmosphere. But there's not a lot of model to predict how much of this is moving from here to there and to the different domains. We've only used models for other chemical classes to models developed for other chemical class, but whether they are applicable to VOC, we don't know. So with these studies, the question is, are those models applicable for VOCs? To a bigger extent, no. Only for a very small fraction, we can say, yes, you can apply this. So here's the result of the studies. These are the different models that are currently in the literature about those partitioning data, and that this is our predicted experimental concentration. And when you look up here, so you have 2.27, you have 785, 321, so they're close enough, right? But then when you look at butanol, that's just massive difference between what is known in the lab and what is predicted by those models. And especially for this compound, there's just this, it's not going to work. So the reasoning is that for like butanol, there's a massive temperature dependence, and not going to get into the details of this temperature dependence, but what it essentially says is that only compounds that are near, that are close to the properties on which these models were developed, you can apply them. Otherwise, for VOC, they are not useful in any way. So this is just a short, what do you call this? Description of what am I going to, my research interest in the future. So right now we're working on, not working, but writing a proposal on permafrost thawing. And the thing is, we want to relate VOCs and to a changing climate. So how do VOC emissions affect, for instance, climate change? Now for this research, we know that the permafrost in Alaska or in the Northern Hemisphere, they're thawing. So this is supposedly ice, but now even in late fall, they're started to thaw. And once they thaw, you have very rich nutrient here because of fungi and bacteria munching on those organic compounds. And those bacteria can leach into the permafrost layer. And then whatever organic compound is in the permafrost layer, they would munch on it and then emit volatile organic compounds. Now, once they emit those gases, they form pockets here. So these are these growing holes, cartoons here. So, and then they form these small holes, and finally they may become larger and the ground collapse. So that's a big problem in Alaska and in some places now, because the roads are buckling because of the permafrost thawing, aside from heating of the surface, and they're collapsing. So we would want to predict using a combination of metagenomics and volatilomics to determine, can we predict where we can build structures that might be safer and might be more stable. So the other one is the impact of climate change on harmful algal blooms, especially on the point of view of aerosol, is that there's a massive steady increase in harmful algal blooms over the years that correlates with increase in temperature, and that there are reactions in particles here that would actually occur. But if you have seawater, for instance, and you have toxins there like bradytoxins, they're not going to undergo transformation. But once they become aerosolized, and some of them do, because ocean waves are forming high amounts of free of particles as the water waves actually breaks into continental shelf, they produce these reactions. And from those discussions I've mentioned, we have acid catalyzed reactions occurring as a consequence of those reactions. And there are photochemical reactions happening on the interface. And on the perspective of aerosol, the aerosol is fascinating because there are surface reactions here that are very interesting, and there's a lot that has not been known. So what this would do is that if you have a toxin and you have ozone, which is a lot, there's a lot in the atmosphere depending on the pollution level of the city, it can make those compounds break. And once they break, they are being transformed to something else and fragment to something that might be more toxic or less toxic. So overall, there's this transformation of this compound that would not normally happen in bulk seawater or in the lake, but in the atmosphere, they would react. So with that one, thank you for your attention and to those who attended, and thank you so much.
Okay, thank you very much, Eric. That was a very fascinating talk. Some of the things that we don't really think about and exactly affecting a lot of us, particularly all this particulate matter that make up the polluted environment. So now we're open for questions. Does anybody have any questions to ask our speaker? Here's a question from Gladys on the chat. Eric, given that exposure to nanoparticles could induce the production of reactive oxygen species, which is a predominant mechanism leading to toxicity, for example, excessive ROS production causes oxidative stress, inflammation, and subsequent damage to proteins, cell membranes, and DNA, do you think that there are significant health effects to humans from long-term exposure to metal-based nanoparticles?
Yeah, so there's a lot of I think metal has always been known to be very hazardous, and a lot of them because they can participate on those phantom type reactions. So over the course through chronic exposure, indeed that's a lot of a big concern. So, and hopefully a lot of incinerators have air pollution control devices that can remove this particle before they are being emitted into the atmosphere. But I think in the Philippines, we don't have a lot of that one. So, so that's a big risk factor in terms of health due to ROS, which are catalyzed by metal species and not just from the EPFRs alone or from other pollutants. So,
yeah. Would changing materials that are used for nanoparticles be useful in preventing this from happening or diminishing this type of effect?
It can, because some of these organic compounds are not normally very active, unlike transition metal oxide. So in the research that we're doing on those free radicals on zinc oxide and titanium dioxide, we're also working on carbonaceous nanoparticles like graphene and fullerene to see if they form EPFRs. But to some extent, there's been some studies that link generation of ROS also from fullerene. So,
is that?
yeah. So I haven't read anything about graphene producing ROS, but for EPFRs in the perspective of whether they can form, we're still working on that one. So the student, I think this evening measured some studies about that to see if we can generate EPFRs on surfaces of a clean carbonaceous nanoparticles. So.
Yeah, yes, I think it's a very interesting, particularly since there are a lot of studies that are trying to develop new non-toxic and biodegradable nanoparticles for delivery, particularly of a lot of drugs, for instance, targeted therapies in cancer that's being studied. There's another question here from Ruel Nacario. What is the possibility that of the permafrost thawing releasing potentially harmful disease-causing organisms?
Yeah, so that's one concern with this proposal ongoing is that we have to be careful with sampling because we have a virologist on board on writing this one, and part of it it's not really the focus now, but I think there are unknown microbes or pathogens that you might resurrect along the way, and if we're sampling. If in the event this one gets funded and you sample this course in Alaska and bring here in Georgia or USC, then you're like spreading pathogens that we don't know of. So I think that's one concern and we really need to address that one. So, and I was, before this presentation, I was reading on it's a good study though, because they found that there's a pathogen, there's a bacteria, Saccharomyces, which actually produces strong antibiotics that would work to counteract bugs. But these places have unknown pathogens or beneficial microorganisms that would be useful, but in terms of the risk of bringing this out, I think that's a very big concern too.
Yeah.
So,
Can they, I mean, I know there's a lot of studies with microbiomes, for example, with this, are you collaborating with a lot of other scientists that are looking at microbiomes on surfaces, particularly in these areas that are being revealed by melting of the permafrost?
yeah, so I'll be collaborating with a person at UNC Charlotte. So she did a bioinformatics there. And
Mhm.
also I'm actually, since we'll be sampling, we might as well just send it to some commercial genetic sequencing facility.
Right, yeah.
So we
Mhm.
actually know Easy Biome in Maryland, and they can do metagenomics and metatranscriptomics because we want to assess, the mechanism in which these organic compounds are being oxidized, fermented, and then emits VOCs and methane and CO2. So
Very fascinating, because I know that there are people in our department as well who are actually looking at microbiomes on the surface of the ocean for a lot of the marine science studies.
cool.
Yeah. Do we have any other questions from the audience? Okay, here's a question from Clemton Shan Fronda. Do you think that nanoplastic particles are also something to worry about in terms of public health exposure, especially on a long-term exposure?
Yeah, it is a very concerning one, and I think right now, just looking at the funding atmosphere, there's a lot of I think funding for a nanoplastic. So first of all, it's not supposed to be inside our body, so that's already like low hanging fruit, right? We need to understand that, and that I think the very important question is how do we remove these nanoparticles and from the environment in which we're not supposed to eat them in fish. But in terms of a long-term exposure, I think because those nanoplastic would absorb some metals too, and then they might be catalytic, and they are decorated with some corona on the surface that might render their toxicity a lot more toxic than other plastic. But most nanoplastic, although they're nano in terms of the terminology, but they're still not really nano, in which how we define nano, they're still like big. So yeah, but there's still a lot of research on nano, especially standards on measuring nanoparticle, which can be very confusing.
I guess my question then is, since there are a lot of nanoparticles that are being incorporated into everyday materials that we're using, is incineration or just throwing them in the dump the main way of disposing of them? Or have people actually developed materials and looked at studies as to the safer ways of disposing of them? I know the US Army funds a lot of these kinds of particles for chemicals and waste that they have, but I don't know if they're doing that for a lot of the new chemical material, nanoparticle materials that are being developed.
Yeah, well, in the United States, there's really no rule on what can be disposed in incinerator. So most of this, or even what can be incorporated, unlike in the European Union, they're usually ahead of the United States in terms of legislation with new contaminants or new materials. And I think it's only silver who has some, how to dispose it, but for other materials, not so much, unless you're working in the university in which you need to dispose them properly. But otherwise, you can buy. I've heard somebody, when she went to Dubai, got some coffee and they just sprinkle some nano gold, whatever. So like fancy coffee with some gold in it. Like, why do you drink that? Are you Michael Faraday? So anyway, in terms of that one, there's not a lot of legislation in the control of the disposal of nano.
Because most of them are silicon, but then also a lot of them have been using gold material, gold as one of the bases for the nanoparticles.
Yeah.
And they've been going away from that.
And titanium, like this wall, has a lot of titanium dioxide. And also, I forgot to mention that titanium dioxide, if you like Krispy Kreme, I don't know if they change it now. The white stuff on your donuts, they're not sugar. They're titanium dioxide nanoparticles. Or those in soy milk, or those in soy milk, they're actually, they add titanium dioxide there to change the color and make it more, I think, inviting. So, but that's an old research, I think Paul Weir from Arizona did a lot of ICP studies on how many consumer food items actually contains nanoparticle, and there's a lot of them. Toothpaste has a lot of titanium dioxide. Now, the thing about titanium dioxide though is that if you ingest it, it's not very hazardous. So, but if you inhale it, it's carcinogenic. So in the European Union, they banned already titanium dioxide incorporation in food products, and not here in the United States. So just be aware that there are products there. If they're like white, maybe they might have titanium dioxide, and you don't want to eat what is in your food and what is on this wall.
Just read the labels on the food to look at the ingredients and what's
Yeah.
present in them.
Yeah, and I think
Yeah,
maybe
okay.
I've not checked it, but they might have updated the project on emerging nanotechnology at the Woodrow Wilson Center. So that
Yeah.
would be a good database for you to look at if you're
Oh, I see.
concerned. So
Okay. Well, that's good to know. Okay, anybody else has questions? Okay, so with that, I just want to thank Eric for a very fascinating talk, very informative with respect to aspects in pollution as well as food additives. And so with that, let's give Eric a big hand. Thank you so much, Eric.
thank you.
Yeah, and we hope to invite you in future talks, and hopefully you'll present some of these at the APAMS.
Yeah, I think
there's
it coincides with the dioxin conference in New Orleans.
There's another one from the chat from Gladys. Eric, please share the link about titanium oxide incorporation in food, if you could.
Okay, yeah, I will. So
Okay, yeah, okay, great. Thank you so much. All right, good.
thank you so much.
Thank you everyone for attending. Mario, did you have anything else?
No, can we have like a photo? A group
Oh,
photo?
everybody show your faces.
So I think we should unshare. Eric should unshare.
Eric, we can turn off your slide now and ask everybody to open their cameras so that we can take a picture.
I can't find it. Sorry. Okay, there.
Stop sharing.
Okay.
Okay, so can we ask everyone to open their cameras so that we can take a picture? Okay. I'm trying to open my camera. All right. Okay, everybody smile. Say cheese. Okay, ready? One. Come on. Some people still haven't shown their faces. 1, 2, 3. One more. 1, 2, 3. Okay, thank you everybody for attending tonight's webinar. Okay, somebody else showed her face, so I'm just gonna take one more photo. Don't go away. Okay, ready, set, go. Okay. All right. Thank you very much, everyone.
Thank you, everyone.
Seminar. And don't forget, next week we have another fireside chat, a very interesting one, and hope to see you there again. And so please keep in touch with all our future seminars and try to send some abstracts for the APAMS meeting. Okay. All right. Good night and good morning.
Good night.
Are you gonna show the video?
Yes. Yeah.
Oh, use a video from Mario if you have time.
There's sound now.
The one.
There's a sound now. Yes.
There's sound now.
Double down on our efforts to catch up with science and technology and innovation. science and technology and innovation, these are indispensable in the modern economy, in the modern world.
The problem being that, while we have our PhDs here, there aren't enough of us. And everywhere else in the world, for research to be successful, you need a complete research group.
It's very important that the PAASE members and scientists based in the Philippines are continuously in contact, so we will know the needs of the Filipinos and opportunities that the Filipinos can have to do science with their PAASE counterparts.
I think that is one of the good programs that they keep on supporting some of the initiatives of our researchers and scientists here in the country.
The role of this cooperation among these government agencies really is to be able to attract science and technology global-based companies to set up shop here. And that is really key in terms of developing the science and technology innovation ecosystem in the country.
We really need to ratchet up the appreciation, the recognition of the importance of science and technology and innovation.
And we are all trying to attain what we call the common good, the greater, higher, long-term good of the Filipino people.
Thank you.
Okay, thank you. All right,
Thank
good night, everyone. Good morning. Have a great day. The Filipinas.
Salamat,
Okay,
Eric.
thank you, guys.
Thank you,
Thank you.
Eric.
All right,
See
and see you.
you again soon.
Okay.
Good night. Bye.
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 Get well, Gladys.
Yeah,
Yes,
get well.
thank you. Bye.
Okay.
Bye-bye.
Bye.